Hello, and welcome everyone to this podcast for genetics. In this podcast, and in the next podcast, we're going to focus on the phenomenon of meiosis, and why it is so important in understanding the basis of genetics. Remember, at the beginning of the semester, seems like a very long time ago, we began with a fairly robust discussion of DNA. We talked about the parts of DNA, how DNA was the genetic material. We talked about how DNA copied itself through replication. We talked about DNA as the genetic material. And we also, of course, talked about other features of DNA, how it was packaged into chromosomes, and a lot of other things. And then as we moved on to the material for test 2, we talked about how we take this DNA and we ultimately will generate some kind of trait. And we did that by first making RNA molecules. We talked about many types of RNA molecules, but that all RNA molecules were made through a process of transcription. We then talked about how that RNA molecule could often be used to make a protein. And we talked about different forms of protein, but ultimately the take-home point here was that these proteins are what provides traits for the individual who had these pieces of DNA. And the process of making these proteins, I should mention or remind you, was translation. And then the last part of the material for the last exam, we talked about how this whole process here was regulated. And if you remember, way back earlier in the semester, we mentioned that this whole process of going from DNA to RNA to proteins was collectively called the central dogma, which leads us to this unit's material. The first couple of days, we're going to talk about how we're going to take this DNA and we're going to package this DNA into gametes through a pathway or pathways called meiosis. And that how this process helps generate great diversity in populations. Following this discussion, we're going to talk about how through meiosis and other processes, we generate mutations. And how these mutations can lead to new traits, how they can be a driving force for evolution, and how they can cause diseases. And we'll focus on cancer towards the end of this unit. So let's come back here and focus on how that DNA is going to be packaged through a special kind of cell division of meiosis to produce these diverse gametes. But before we jump into the specifics of meiosis, I want to discuss briefly why is meiosis important. Meiosis is important for many different reasons, and I'm going to just mention 4 here. It's important for us to understand inheritance, which we'll come to during our 4th unit of material for this class. It's also important to understand mutations, which we'll talk about in the next few days. It turns out that during meiosis, we can generate several kinds of mutations. These mutations can, of course, cause problems leading to diseases or death, but they can also be driving factors for evolution. These mutations, as well as just the general process of meiosis, can lead to diversity, or rather, they will lead to diversity. Meiosis explains the similarities and differences we see in populations. And so what I'd like to do is look at a few populations real quick to help explain why meiosis is important in generating this diversity. In these 5 pictures here, you'll see 5 different populations. And remember, a population is nothing more than a group of the same species living in the same community. And what we see here is a population of gators, a population of garlic plants, a population of sardines, a population of ladybugs, and a population of monk parrots. In each of these populations, as you look at them, you will say they look fairly similar. But we know that even externally, we can see a few differences. We can see some differences in these birds and the shading of the green and the white, how much blue they might have under their feathers. These sardines, even though it's harder to see, there are differences there. Ladybugs is a little easier to see the differences, but you also notice a lot of similarities. Same thing with the gators here. They look quite similar, yet there are differences. And we also have to remember there's a lot of differences we can't see that are within inside of the organisms. Meiosis also helps explain the gene pool I swam out of and continue to swim in to this day. You can see here, this is me here, my brother Bob, my sister Tony, and my brother Tom here. And you'll see my dad, right here and my mom right here. And then you see various other members of our family, our wives and husbands and our kids. And as you look at this mess here, you'll say they all look fairly similar. But you'll notice some striking differences as well. These similarities and differences are explained by meiosis and the rules that govern meiosis. So now what I'd like to do is move back to the whiteboard so we can begin a more in-depth discussion of the mechanism of meiosis and how meiosis is going to lead to an explanation of what I lovingly call this genetic mess. As we begin this, I want to begin with versus mitosis. But I think it's important to give you a brief reminder here. And let's begin with a fertilized egg, which we'll call a zygote, a one-celled embryo. And just to keep it relevant to us and what we're familiar with for right now, let's think about humans. This fertilized zygote has 46 chromosomes. We know that this zygote will divide. It will divide many times. And when it divides, each new cell will have 46 chromosomes. And this process of division here is called mitosis. These cells will continue to divide until ultimately a whole organism is produced. This one cell becomes billions of cells. And what's important to note here is that this all occurs by mitosis. And more importantly, I should stress, the cells within this organism that are somatic cells are the product of mitosis. The key to mitosis is that every somatic cell that is produced, from the very beginning to the end, are genetically identical. Gene regulation helps explain how these genetically identical cells behave differently. But every one of these cells has 46 chromosomes, and they have the same genes, minus a few mutations here and there. But now, let's come back to this zygote. Where did this zygote come from? Remember that these zygotes came from the fertilization of an oocyte by a sperm. These two each contain 23 chromosomes, so that when they fertilized each other, they were added together to produce the zygote with 46 chromosomes. But where did these sperm and oocytes come from? And remember, sperm and oocytes, generically, we call these gametes. Where did they come from? Well, as you know, the sperm came from the father, and the oocyte came from dear old mom. And they generated these gametes, sperm and oocyte, through a process of meiosis. Remember that each of the cells in this parent had 46 chromosomes, and each of the cells in mom had 46 chromosomes. The gametes have half that number, and that's because of meiosis. Meiosis, as we will learn, generates the diversity that we ultimately see in the progeny. Through this awesome special effect, you can see that the parents look different than the child, yet you see some similarities as well. And that's because of the diversity generated through the pathways of meiosis. Let's now focus exclusively now on this. And let's begin with talking about the two parts of meiosis. Meiosis will begin with a diploid cell. Diploid cells we often abbreviate as 2N. In humans, that means we begin with a cell that has 46 chromosomes, or 23 pairs of chromosomes. This diploid cell will now undergo meiosis I. We often call this first stage of meiosis the reductional stage. It's reductional because it reduces the number of chromosomes in half. So these cells at the end of meiosis I will each have 23 chromosomes. I drew them in a different color because what we also know is that the diversity that is generated in meiosis occurs in meiosis I. So let's just write generates diversity here. And we'll come back to this in just a moment. Now the second part of meiosis is meiosis II, cleverly named. Meiosis II is often referred to as equational division. And that's because each one of these cells divides to produce two cells that are chromosomally the same. Equal number of chromosomes in this cell to its two cells that are produced. So it's called equational. This part of meiosis II, is similar to mitotic mechanisms. In the rest of this podcast, we're going to focus on Meiosis I. But real quick, I want to make a short list here of where this genetic diversity is generated. And we'll come back to this as we go through the podcast. One is crossing over. We'll talk about this when we talk about prophase I. The second part of genetic diversity is due to the random assortment of chromosomes. And we'll talk about this in more detail when we talk about anaphase I. And the third part isn't really linked to meiosis, but we'll call this random fertilization. Which sperm is used to generate the zygote and which egg is used to generate the zygote are fairly random. And so because of that randomness, we generate diversity. All right, let's now talk about cell division with meiosis. Remember that the cell cycle has various stages. It has G1, it has the S phase, it has G2, and it has the M phase. And for us, this M phase is meiosis. We're going to focus on meiosis, but I think it's important to think about where that cell came from that is going to be used in meiosis. And to do that, I'm going to draw a cell here. And I'm only going to draw 2 pairs of the chromosomes. So this might be called chromosome 1, and these might be called chromosome 2. Remember, every diploid cell has a homologue of every chromosome. One of these chromosomes came from one parent initially, and one came from the other. Same thing with chromosome 2 here. I drew them different sizes just so it would be easier to follow them as we move through the cell cycle and meiosis. Now remember, as it exits G1 and enters S phase, each of these chromosomes are copied, like so. Just to remind you, over here we say that there are 4 chromosomes. We would also say there are 4 pieces of DNA, or you might say 4 chromatid. Over here we still have 4 chromosomes, because as long as the copy DNA is attached to the original one, we call it 1 chromosome. So still 4 chromosomes. But we would say there are 8 pieces of DNA, or 8 chromatids. Remember that we call these 2 sister chromatids. This blue one and this red one are homologs, but these 2 blue ones are sister chromatids. They are identical to each other because it just copied it. These 2 are sister chromatids, these 2 are sister chromatids, and these 2 are sister chromatids. Don't forget that. Sometimes it's easy to lose that. After the DNA is copied, it enters G2, still with the DNA copied as such, and now this cell is ready to enter M phase. And that's where we're going to pick up this cell as it enters M phase. Oh, and also if you're keeping score at home, there are still 4 chromosomes and 8 pieces of DNA, or you could say 8 chromatids in this cell too. We're going to follow those numbers as we go through meiosis, so keep that in mind. During meiosis I, because remember that M phase is going to be a combination of meiosis I and meiosis II. So remember in this podcast, we're only going to talk about meiosis I, even though M phase is a combination of meiosis I and meiosis II. We'll cover meiosis II later. Meiosis I involves 4 different stages: prophase I, metaphase I, anaphase I, and telophase I. And at the end of meiosis I, we'll end up with 2 cells that look something like this, where each cell will get 1 copy of these, of one or the other, that is, and they'll get 1 copy of this smaller one. Now this, and we'll come back to this when we talk about anaphase I, but that's where we can generate some diversity, because this cell here could either get the red one, the smaller red one, or it could get the smaller blue one. It could only get one of them, though. So I'm just drawing it this way. But remember, I could have equally have drawn this where it got the 2 blue ones here and 2 red ones here. The key is that each one of these should have 1 copy of chromosome 1, 1 copy of chromosome 2. Only half as many. That's why it's called reductional division. Now let's talk about each of these phases. And let's begin with prophase, middle and late prophase. For us, I just want to collectively put it into prophase 1. And we'll talk about everything that happens during those different parts of prophase. So remember, what we had in that earlier cell here is that we had our 2 chromosomes that had been copied because it's gone through S phase, right? So let me redraw those. Now, what I didn't show on the previous slide, and we often omit in the drawings when we talk about G1, S, and G2, but it's important to remember that That all occurs in the nucleus. So let's draw the nucleus here. And it's important to draw the nucleus because during prophase I, one of the things that happens is the nucleus breaks down so that essentially the chromosomes are in the cytoplasm now. So let's write that over here. One of the hallmarks of prophase I is that the nuclear envelope, or you could just say the nucleus, but really it's the nuclear envelope, breaks down. Though it's hard to draw this, you should also know that just like in mitosis prophase, in prophase I, the chromosomes condense. They form that very tightly wound up, structured chromosome, which is essentially unusable to turn genes on or off in, but it's ideal to make sure that chromosomes get assorted properly during meiosis. The next thing I want you to know is that this very important process called synapsis occurs. And what occurs during synapsis is that homologous chromosomes line up at different regions. They interact with each other. During synapsis, a very important thing occurs, and that is crossing over. One thing we should point out is that crossing over between homologous chromosomes occurs during meiosis I, during prophase I. But it doesn't just randomly assort everything. It only occurs at a few points. And as a result, what will happen is you may end up with a cell that looks something like this, or rather I should say chromosomes that look something like this, where there has been an actual physical exchange of genetic material across these chromosomes. Now you might think that's not a big deal because these are homologous chromosomes. They have the same genes. But it's possible that this blue one at that region there had a gene that was mutated. So I'm just going to draw a minus there. This is minus on both of them. And this one here was plus on both of them. Not mutated, a specific gene, that is. But now what we've done is we've generated these sister chromatids that now have a mutated gene on this chromosome and a non-mutated one here, and likewise for the red one. It could occur on the smaller chromosome too, but for simplicity, I'm just not going to draw it. But remember, this is one way that we generate a lot of diversity during prophase I because of this crossing over between homologous chromosomes. It never occurs between non-homologous chromosomes. It only occurs between homologous chromosomes. Another term I want you to know, because it's related to synapses crossing over and crossing over, is this term called chiasma. And the chiasma is nothing more than the physical point where these two chromosomes synapse. So the chromosome synapse, so during synapsis, this chiasma is formed here at the physical interaction between these two so that crossing over occurs. And the very last thing I want to talk about with prophase I are the centrosomes. If you remember from mitosis, the centrosomes are found in the cytoplasm. And that's one of the reasons the nuclear envelope has to break down. Because once it breaks down, these centrosomes can now migrate towards opposite sides of the cell. And that'll become important in the next phase, Metaphase I. So let's move to Metaphase I. And I'm going to leave this cell drawn here, just so we can remember where we were at the end of prophase I. I'll just write P1 for prophase I. And now we want to talk about Metaphase I. During Metaphase I, what will occur is the aligning of those chromosomes. So I'm going to draw this cell a lot bigger just to give me some room to draw. If you remember from mitosis, there is this area here that we call the metaphase plate. In mitosis, well aligned on opposite ends of this metaphase plate were sister chromatids. What's going to align along the metaphase plate during metaphase I are homologous chromosomes. But let me draw it so it's a little bit more clear. So this chromosome here will be on one side, and remember, we can't forget that crossing over has already generated that one level of diversity. And over here is the homologous chromosome. And over here is the homologous chromosome, also with that crossing over product there. Now, which side of the metaphase plate, this red one or this blue one, line up is completely random. I could put the small red one here or the small blue one here. I'm going to put the small red one here for no particular reason, because it's random. There would be no reason. Now let's think about why it is that these chromosomes lined up along this metaphase plate. It wasn't by accident or by some kind of magical spell, but rather these centrosomes here are microtubule organizing centers where they send out these special microtubules that we're going to call spindle poles. And it's on either side here. And so they attach to these chromosomes as it transitions into metaphase and extend them and retract them in such a way so that each homologous pair of chromosome is on either side of this metaphase plate. Again, it's important to note that it's completely random as far as which homologous chromosome is on either side. And that's going to be really important as we move to anaphase I. And that's all I'm going to say about metaphase I. Okay, so let's now extend from this metaphase I cell and talk about anaphase I. So let's draw our cell here. We can draw a metaphase plate again. And let's draw our centrosomes over here. And now what's happening during anaphase I here is that the centrosomes that were formed up here in metaphase I are going to begin to retract and pull back. And as they retract and pull back, they will pull with them the homologous chromosomes that they're attached to. And even though in metaphase I is how they line up here, the actual diversity we say happens in, the actual diversity causing event happens in anaphase I, because that is when the chromosomes are pulled apart and arrive at these ends of the cells. Now next I want to talk about our final stage, and that is the stage of telophase I. And during telophase I, essentially what's going to happen is that within this cell here, the cytoplasm will now divide. Draw this a little different here and show how during telophase these new membranes form to generate these two new cells, much in the same way that telophase occurs in mitosis. So you end up with these two cells that contain one homolog of each chromosome. Now the cell is ready to move to Meiosis II, and ultimately after Meiosis II to form a gamete. So remember, we started off with a cell that looked like this at the beginning of meiosis. And now we've ended up with two cells that look different. Instead of having 4 chromosomes, 2 copies of each homolog, we have 2 cells with only 1 copy of that homolog. In addition, we can see that this homologous chromosome is a little bit different than it was before because of crossing over, bringing in that 1 piece of DNA from the red one. And likewise, the red one is also different for the same reason. Another difference we see is because of anaphase I. Instead of having 2 blues and 2 reds, we have 1 blue and 1 red, and 1 red and 1 blue. So again, more diversity is seen. And once these gametes are produced and used, that will generate the final step of diversity during random fertilization. The last thing I want to talk about are these proteins called cohesin, separase, and sugoshin. Okay, as we talk about these, I'm going to draw the cohesin protein in purple. I'll draw the separase protein in green, and the sugoshin protein in pink. And where these are important, at least initially, will be in meiosis I. So I'm going to draw a cell here in prophase I. In prophase I, we'll have our 2 homologous chromosomes, and I'm only going to show one of our homologs here. Now, a couple things I'm going to tell you about cohesin here is that, even though we didn't talk about it before, it holds the sister chromatids together. So this cohesin originally forms during S phase, again, to keep the sister chromatids together. But they also do something else. These cohesins will allow these 2 homologous chromosomes to interact with each other to form that chiasma. Remember, we talked about that earlier. So these cohesins will allow this physical association between these, I'm just going to draw it like this, it's a bit exaggerated, between these homologous chromosomes. Because the cohesin proteins, in addition to holding 6 chromosomes together, will hold these homologous chromosomes together. Now, during anaphase, remember what happens is that we have one of our homologous chromosomes on this side of the metaphase plate, and one of ours on this side. We might as well be consistent and draw the crossing over event. Now, what I didn't show you before, when we talked about this, were the cohesin proteins holding them together, and holding them together here too. So what happens during anaphase, so that the centrosomes can pull these apart, is that these cohesin proteins have to be removed. If they're not removed, then you can't pull these 2 apart. And so that's where separase comes in, an appropriate name. Because what it does is separase will come along here, and it will degrade the proteins, the cohesin proteins, holding these 2 homologous chromosomes together. Now, with the cohesin removed, we can pull the two homologs apart from each other. Now, what you're probably asking yourself is, if separase is degrading the cohesins apart to pull the homologous chromosomes away from each other, what's preventing separase from degrading all of the cohesin, allowing the sister chromatids to come apart? Because at this point here, and at the end of meiosis I, the sister chromatids are still held together. And they are still held together because cohesin is still there. But why is cohesin still there? It's because, as the plot thickens here, the protein sugarshun, which is a lot of fun to say, wraps around these cohesins to protect them from degradation by separase. By sugarshun protecting cohesin, remember, holds the sister chromatids together, but allows the homologous chromosomes to be pulled apart. We'll learn, as we move to meiosis II in the next podcast, how the sugarshun will be removed, allowing the sister chromatids to be pulled apart. However, you're going to have to come to the next podcast in order to see how that story unwinds. That is all I have for this podcast. So if you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye.